Hello, everyone. Welcome back to the Healthcare AI Forum. And today, I'm really excited to introduce our friends at UCLA. And I will start with Dr. William Xu. He's a professor of radiological science and bioengineering at the University of California, Los Angeles. He's a biomedical informatician focused on maximizing the utility of multimodal data to detect and treat cancers earlier. He's the deputy direct editor of Radiology AI, and he's going to present along with his two students, Noor and Stephen, a special session on medical imaging. And also about me, my name is Yuan, host of the Healthcare AI Forum. So now, without much further ado, let me turn the stage to William.
Great. Well, thank you so much, Juan, and everyone involved in organizing this series. Really appreciate the opportunity to come and to present. And I think I understood that you guys have already done some journal clubs or sessions like this previously. And so really happy to be part of this to contribute a perspective on medical imaging. So my hope today is that, first of all, we will give you guys a nice survey of the current work in the space of medical imaging. It's very active, as many of you may already know, in the area of AI and using AI to transform how radiologists interpret images. And one of the things that we want to highlight here is really some of the diverse work in trying to extract biological information from images. So the papers we chose, as well as the students' presentations, will really revolve around this theme, and that is a core aspect of our research. So before I get into my paper, I just want to introduce the group. So this is the Medical and Imaging Informatics group, specifically my lab, the SHU lab, that is one of several groups in this, group. And really our mission is to re-engineer healthcare by transforming data and knowledge to improve all lives. We are located at UCLA, actually just right across the school, the street from the School of Medicine, and UCLA is a single campus. So we really have all the engineering and applied sciences type of expertise all in one place. Now, many of you may already be working with multimodal data, and certainly our lab is very interested in this space. As you know, there's a lot of data that's available in the electronic health record, spanning from the record itself all the way to images, digital pathology, genomics, wearable sensor data, familial history, all of these different data points that are now actively being collected. And one of the things that we're very interested in using this data is to look at how do we apply or at least correlate some of the information from other biological scales and other modalities to help us understand images. And that could be looking at the relationships between radiology and pathology as a direct comparison, but also looking at how these image features may be related to gene expression and disease severity. And so the core interest of my lab is really to look at if we take an image, so this is, for example, a cropped image of a CT of a lung. And so there's a lot of patients who are now undergoing lung cancer screening, and maybe at their baseline screen, they have nothing abnormal in the image. But then over time, they may start developing a nodule. And so the imaging captures this, and we can see these changes over time. And the question is, can we use the image to help infer some of the biology? As many of you guys may be aware of the hallmarks of cancer. So certainly looking at all of the different functional and changes that occur on the molecular genomic level that ultimately may relate to this nodule being cancer. If it is cancer, if it's an aggressive type of cancer, and ultimately what may be the best type of treatment. And so my lab and some of the students here work on various aspects of this process. And so this is actually a review paper that we put on archive. Stephen's actually one of the co-authors. And one of the things that we talk about is really the challenges around structuring this data, both clinical imaging, molecular data, and really trying to build these multimodal models, but not only that, longitudinal multimodal models so that we can capture these changes over time and to apply these to different tasks like diagnosis, subtyping, and other types of downstream questions that we may have. So these are some of the example type of things that we do in my lab. Feel free to reach out to me if you have interest in the space. Would love to find collaborators as well as others who work in this area. So would be happy to connect. So with that, I'm just going to briefly introduce some of the talks that we will have. So it's going to be a bit of a potpourri of different talks. It just so happens that all of these papers came from Nature Communications, but that was really by accident. But really, we chose these papers primarily because we have some interest in our lab in some of these topics. And so the hope is that we can present a high-level overview. Please do ask questions. I know this format is not always with the webinar. It takes a little effort to do some interaction, but we will collect your questions and we will try to have an interactive session at the end where we will try to answer it as sort of like a panel. But definitely please do pose questions about the papers or about the research in our lab. So I'm going to start by talking about this paper that was published a few years ago, but it was a very interesting one in trying to use medical images to predict biological age. And so the title of the paper is Using Deep Learning to Predict Abdominal Age from Liver and Pancreas Magnetic Resonance Images. It comes from a group at Harvard University. And I would say it's probably one of the large, and it wasn't necessarily the first, but it was definitely one of sort of the precursors to some of the work in this space where we're now seeing a lot of interest in using the large quantities of medical images to try to do this type of prediction. So for those who are not familiar with the concept of biological age, as we all know, we go through biological changes as we age, right? And so certainly at the molecular level and at the phenotype level, there are changes as we age over time, and some of that can be visualized both in medical imaging, in other ways. These include architectural changes, vessel dilation, splenic growth, fibrosis, muscular atrophy, and so our organs naturally go through change, not just over time, but also from factors like our lifestyles and other environmental exposures, et cetera. And so we know that aging contributes to many diseases, and certainly could be a shared root cause for these conditions. And so one of the things that we are particularly interested in is, well, can we use imaging, just like how a lot of people are using molecular data, to predict this concept of biological age? And so sort of compared to chronological age, which is the time since an individual's birth, really the biological age is supposed to be capturing the state of the body of an individual at that time. And so how this typically is done is you have some population sample, so have a large cohort, normal patients, for example. Typically this was done originally in genomic and blood tests. Steve Horvath, who was actually previously a faculty member at UCLA, was one of the key drivers in this area, where you would do genomic analysis, extract sort of information that you can then do some type of statistical learning, where you're essentially associating gene expression to age. And so you can create a linear model like this, where you look at, minimizing the error between the predictions of age given this information. And you can start seeing, for example, if you predict an age that is greater than a individual's chronological age, then you could potentially call them fast aging, versus if they are lower than what was the chronological age, predicted age was lower than the chronological age, then maybe you have some kind of slower aging, right? And some of the hypothesis around this is that if you have faster aging for an organ, that it potentially could be a sign of disease or some type of risk that potentially could be translated into something actionable. And so again, as I mentioned, there's a lot of different studies, primarily in the omics space, that try to do this type of prediction and to predict chronological, or I should say, or characterize this biological age. So in this particular paper, the authors were trying to develop and validate an imaging-based abdominal age predictor, they call it AbdAge, by training a deep convolutional neural network to predict age from liver and pancreas MRI images. And so hopefully you can see the relationship between why I'm choosing this paper and the work that we're trying to do in my lab, where we're trying to, again, tie the imaging data to some kind of biological evidence that ultimately could be a predictor of risk. So how they did it in their work is they used a very large data set, in this case, the UK Biobank. So this is specifically a study that's happening in the UK. They have 500,000 de-identified participants in the UK between 37 and 74 at the time of enrollment. And out of that, there was a subset of them that are undergoing a series of scans. And so here, at the time they did this study, there was 44,000 liver MRIs and 36,000 pancreas MRIs. And what they wanted to do was take this data and try to train a imaging-based classifier for biological age. Just to note a little bit that for those of you guys who are in the medical imaging space, certainly in these type of studies, there's standardization in how the images are acquired. So there was a very specific set of protocols that were used for all of these participants as part of the UK Biobank when they acquired the liver and pancreas images. Just very briefly at a high level going over the methodology. So as you can see, there was a relatively large cohort of images for liver and pancreas. And what they did was they did some QC on it, removed some images. They converted the images from DICOM, which is the inherent format, into, in this case, JPEGs. They did some data augmentation, again, to help train their models. Then they used essentially two different architectures, the Inception ResNet v2 as well as Inception v3. All of these were trained on or pre-trained, I should say, on ImageNet. They added sort of what they called a side neural network, which is, in order to add age and sex, or I should say sex and ethnicity and other inputs, they basically added that to their model. And then they did a whole set of ensembling because they created multiple different models for variations of images. And then they created these ensembles that then they used to predict their outcome. And so there was 15 age predictions that were generated. So there was 15 different ensemble models. And they ultimately took these predictions and they looked at the relationship between GWAS and what they call XGWAS, which is anything outside of the genetic component. They also did some type of interpret or explainability analysis where they looked at attention max, for example, to just see where the model was focusing on as part of its predictions. So for those who haven't seen pancreas and liver MRIs, this is just an example from their paper that they kind of highlight. And you can see that, the images are processed in some way. There's the raw image that they say here, and then they also did a histogram equalization, which is essentially, as you can tell, kind of trying to harmonize or normalize some of the pixel values so the contrast is a little better within the tissue regions. And just to kind of talk a little bit about their ensembling, so what they did was they actually created 3 models for the liver and then 3 models for the pancreas just on the raw images. And then they also created 3 models for the liver and 3 models for the pancreas on these contrast or histogram equalized images. And then from there, that start here, then they actually combine them. So now they have an ensemble of those models to predict, for example, in the liver MRI, they would do this where they would combine those 6 images. So that would be the organ ensemble. And then they did that for the pancreas as well. And then they combined the pancreas and the liver ensembles to then come up with this general abdominal age prediction. So it's a whole set of models. We'll talk a little bit about, whether that's generalizable, but that is kind of the approach that they took. And the evaluation metrics is that they used R squared as a measure, root mean squared error, mean absolute error. This makes sense because at the end of the day, it's predicting a value for the biological age. And so this is the metrics that they use to try to correlate that with the chronological age. And then confidence intervals. So they basically did a lot of bootstrapping and essentially a cross-validation to try to see how reliable the performance was. And just to give you a sense of kind of the analysis that they did, so they correlated this with other organ systems. So I'm not going to highlight this as a result, but they did try to correlate their age to other age classifiers that exist at the time of their study. And this was using other types of images and other, body parts. They also looked at the genome-wide association studies that was available from the UK Biobank data. And then also the what again they called XY association study, and X essentially means non-genetic measured correlates of aging. And they looked at biomarkers for it was whether sensor biomarkers, clinical phenotypes, diseases, family history, etc. Here's just a summary slide of individual types of models in their group and their performance in terms of R squared and RMSE. So you can see that R squared value is actually relatively quite high, between 0.7 and 0.8. This is for their final age prediction. And then they also then split this out to liver and pancreas. So as I said before about the ensembling, they split the results into individual categories. So for example, this one is just the liver ensemble. And here are the different three models for the contrast or the histogram equalized images and then the raw images. And so you can see that they did that for the pancreas. So these are the 15 model results. And you can see that depending on the inputs, and for example, with root mean squared error, you can see that as you get to the top of the ensemble, it had the least root mean squared error, although, you can see that the error is still between 3 and 4. And then as you get to individual data points, you can see that that error kind of incrementally goes up a bit. And so I think what we're showing here is essentially that this ensembling approach did help them make a better prediction. Here are the attention maps looking at the different types of images. So you can see here that you can see they've chosen some examples of decelerated aging. Again, that's the concept of, the biological age is predicted to be less than the chronological age. Normal aging, which is when it's aligned, and accelerated aging when it's above. And you can see these are for the inherent age groups of, between 50 and younger, or, intermediate or older. And for contrasted images versus normal, or actually I should say these are all contrasted images, but this is for males and females. And so you can see that the model is looking at different parts of the anatomy. And, of course, given this context, it's hard to really interpret exactly what's going on in these images. Some people would argue that this is somewhat subjective to begin with, but I would say that it does give you some sense that the model is looking at specific areas that, maybe of intuitively of interest when you're trying to do this type of prediction. Here's just a quick summary of the genomic data. This is the essentially a list of the genome-wide significant loci that were implicated in accelerated abdominal age, pancreas age, and liver age. So you can see here the individual specific loci that were identified, as well as some of the closest genes. And of course, you can use this information to biologically correlate this to what's out there. And then also you can see here that there's some spatial positioning that you can see where the genes exist on the chromosomes. In this graph, and I know it's probably very small to read, but they're basically looking at all the different categories, the X categories in their X-wide analysis. So these are all, for example, the biomarkers, the clinical phenotypes, diseases, environment, etc. And then here, what they're looking at is the correlation between the liver versus the pancreas age predictions. So what they're trying to say here is that while there is a lot of synergy between those predictions, there are some biomarkers that is related to that are actually completely negatively correlated. So it's interesting to see some of the patterns here of the features. And so, of course, further analysis is needed to see why they're negatively correlated. But ultimately, that was an interesting finding that they got. So if you think that, oh, showing these images that seem relatively similar, there's actually some differences that come out of that analysis. Just to give you a sense of some of the associations that they found. So here you have accelerated imaging, so body impedance, blood pressure, pulse wave analysis, general rating for health, chest pain, breathing, weight loss. So these are all things that they found associated with accelerated aging versus decelerated aging. They had some measures about the hand grip strength. If you did really well on a cognitive test, bone heel dysmetry, in this case, weight gain and no weight change, those led to decelerated aging. So again, they kind of left it at that in terms of their correlation analysis, but further analysis could be interesting to do here. So just to summarize, they developed an abdominal age predictor using these type of neural networks trained on liver and pancreas MRIs. They achieved an R-squared value of about 0.73 with a mean absolute area around 3 years. And abdominal aging, obviously, in this type of data set, there was a lot of different traits that could be associated. And so they did that, and they found 16 associated genetic loci, as well as various genetic, phenotype type of traits that ultimately was interesting to kind of look at for further analysis. And ultimately, they also saw from the attention maps that the model's accuracy is driven by some anatomical features in the liver and pancreas. So some of the things I would just point out as limitations, and these are things that certainly my lab is also very interested in, is number one, ensembling. I know ensembling could definitely help with performance, but that is at the trade-off, honestly, of generalizability. When you're trying to fit this data and you have all these ensembles, to me, a question mark is how generalizable is this? If you have all these models that you're trying to fit, will it really scale? They didn't do any segmentation. The images looked a little distorted. So the question is, the quantitative values that are coming out, how reproducible is it? And as you know, the UK Biobank is a really consistent protocol. So here there's a lot of heterogeneity that could exist in the real world that they didn't account for. And they did not explicitly examine relationships with diseases. So for example, hepatocellular carcinoma could be a risk that you could use this as a risk model. They did not do that. And so that's one open area, is to really translate this into something that could be a risk model for disease. But I think there's a lot of opportunities here. I'm going to kind of wrap my section up to give other speakers a chance. But you can see here that I think we can extend this type of work looking at other populations. And so with that, just want to appreciate again the opportunity, and we'll again take questions at the end. But I will now transition this to Noor, who will talk about segmentation, which is an important problem in the medical imaging space.
Hello. Good morning, everyone. I want to start today by saying happy first year of spring. I'm Persian. We celebrate the first year of spring as our New Year. So if anyone also celebrates it, happy Nowruz. I hope you have a wonderful year ahead of you. My name is Noor. My pronouns are she, hers. I'm a last year PhD student in the computer science department at UCLA. I've been working with Dr. Shu in his lab for the past 6 years. It's reaching 6 years. Today, I'm just going to talk about SAM, or segment anything model, and medical images, and a little future direction on how it's related to my work. And one other work that I've been starting. Let's see, I'll try to wrap it up in 10 minutes. Don't get scared by the number of slides. Introduction. I am not fully sure how many people do work in the medical imaging space, but medical image segmentation is a crucial problem in the clinical practice to get accurate diagnosis, treatment planning, and disease progression monitoring. The current challenges that we have with the models is task-specific models lack generalizability across the modalities. Manual segmentation is time-consuming and labor-intensive. And existing specialist models require training for new tasks. By specialist models is that we have an architecture, they just get trained on one specific set of data sets. So they would be specialist models. And every time for a new task, they have to get trained on the specific data set that they're working on. SAM, or the segment anything model, has shown promise for natural images, but has limitation in medical images. This work is focusing on training the SAM architecture on medical images, so it's called MedSAM. It's a foundation model for universal medical image segmentation. As you can see in the snapshot, it was trained on more than 1.5 million image mask pairs. They have run it through comprehensive evaluation, 86 internal validation tasks, and 60 external validation tasks. It has shown better accuracy and robustness than modality-wise specialist models, and it's able to generalize to unseen tasks and modalities, which, again, as mentioned, is a huge problem, especially in the medical domain for image segmentation. Again, as mentioned, it was trained on 1.57 million image mask pairs. And as you can see, it has 10 imaging modalities and more than 30 cancer types. You can see in the figure how many images for each type of modality of images. We have MR, CT, up to mammography and OCT. Distribution, I think, as again shown in the figure, CT, MRI, and endoscopy were the dominant modalities, but we had ultrasound, pathology, and things that I mentioned. With this kind of diverse imaging modalities, this allows learning rich representations of medical imaging. Let's go to the architecture. Based on SAM architecture. So the architecture is same as the segment anything model that was trained on natural images. We have three components. We have an encoder, we have decoder, and in the middle we have a prompt encoder. The image encoder is the Vision Transformer VITB16 backbone. The prompt encoder is where this model is getting its guidance from. So it has different ways of prompting. You either can choose a point on the image that I want this point or this part of the image to be segmented, or you can choose bounding boxes that the object that I want is going to be inside this bounding box. So for example, we are feeding the bounding box prompts into the prompt encoder. And we have a mask decoder that views image and prompt features using the cross attention and will give us a segmentation. This is a promptable 2D segmentation with bounding boxes, but also processes 3D images, but a series of slices of 2D slices. The initialization, as very common in the field, they've used pre-trained SAM model to still get some information from the natural images before using training it on the medical data sets. The training strategy, it had a fixed prompt encoder. We had trainable parameters, an image encoder of 89 million and mask encoder of 4.1 million. So again, as mostly seen in foundation models, it's a very large architecture with a lot of parameters to be trained. The loss function was unweighted sum of die slice and cross entropy loss. You see the optimizer, the batch sizes, they had really powerful GPUs. So the batch size was 160 across the 20 GPUs, and the training duration was 150 epochs. What you'll see in the results will be the comparison between this model MetSAM with original SAM and 10 different modality-specific unit models. So they have used the unit to train it on or make the unit specialist in the 10 different modalities and the 10 different modalities specific deep lab V3 plus models. For internal validation results, so as we mentioned, they've done both internal and external validation. Starting with internal validation, so the training sets, the data sets that we've already trained the model on. They had 86 segmentation tasks across modalities. MedSAM consistently outperformed SAM. MedSAM often surpassed specialist models, which again, they are supposed to know better based on the specific training data sets that they've been trained on. We saw that SAM struggled with weak boundaries, but performed well on endoscopy images. I think because SAM was trained on natural images and weak boundaries and endoscopy images was harder to segment, MedSAM did a better segmentation on those. And MedSAM showed narrower dice distribution reflecting the robustness that it has across different modalities. Again, definitely because of the many different modalities and cancer types that they had in the data set. So here you see again the dissimilarity coefficient for SAM, unit-specific or specialist, deep lab, and MedSAM. And here you see the dissimilarity coefficient and the different rankings for the blue SAM, the green is MedSAM, unit specialist is purple, and deep lab V3 specialist. And examples of liver cancer, brain cancer, breast cancer, and polyp segmentation across different modalities. And again, I think the important part that SAM is trying to put across is, especially compared to SAM, how on less strict boundaries and the boundaries that are not properly delineated MetSam does so much better than SAM. One of the main criticisms on SAM was how for the unseen images, it really does not perform as well as the unit or the deep lab or any other specialist models. For external validation, they had 60 segmentation tasks from unseen data sets. I hope you don't hear the noise outside. MetSam showed superior generalization. Again, you see the dice similarity coefficient. Specialist models struggled on new data sets. This is widely known in the field of, and one of the main criticism on the models, the deep models or transform models that are trained on specific data sets. For an example, we have picked the nasopharynx cancer segmentation, and MetSam DICE scores was above 87%, and it had over 52% segmentation improvement for SAM, 15% over unit, and 22.7% over deep lab V3. So one of the main criticism over SAM when it originally came out was the fact that for unseen images, it doesn't do well. And MetSam is showing that because of the diverse set of data sets, that they are addressing this criticism and limitation. So it was even performed well on the unseen modalities. Again, more correlative results for external validation that was mentioned. And here we have examples of lymph node, cervical cancer, fetal head, and polyp segmentation across different imaging modalities. That was unseen data and images. For SAM, again, you can see for the different segmentations that, especially around the boundaries, MetSam is performing much better than SAM or the specialist models. One thing that needs to be addressed, especially for, I believe, the foundation models, is the impact of training data set sizes. They did the paper, I think that was a strong point of the paper, that they investigated scaling effect with 3 data set sizes. They tested on 10K, 100K, and 1.57 million, or the full data set. And they were trying to see, I don't know if you cannot see from the figure, the blue box is the 10K, the purple box or magenta box is 100K, and the orange box is the 1 million. What they showed was that the performance adhered to the scaling rule. So when you increase the training data, you also see significant improvement of the results and evaluation. And again, to further prove, I believe one of the criticisms, this is what I think about the foundation models, is that it demonstrates importance of large-scale training. So for any of the imaging segmentation models, it's always hard. We have for the medical domain, we have a lot of in-house and private data sets. And a lot of the papers in Nature and big journals, they publish and they say, oh, the segmentation results that we're getting on our private data sets is 95%, I don't know, accuracy, and we're getting good results. But the main criticism is that not everyone has access to their private data sets, so they can further train their models on. And when you get that data sets and you try to translate it to your own private data sets, they don't perform as well. And MetSam was showing that for, again, as we saw, for the unseen data, it also performs well, but it is also showing that the larger the data set that is trained on, the better performance it will have. So the clinical application, because of the prompt encoder, it's showing that it has annotation efficiency. So human annotation study for 3D adrenal tumors, the 2 pipelines that were compared was manual slice-by-slice annotation and the MetSam assisted workflow, which will first initial sparse linear annotation, then we would use MetSam to segment it, and then at the final level, the expert would refine it. Specialist would sit down refinement. The annotation time was reduced by 82%, and 82.5 for 2 different experts. So it also demonstrates practical clinical utility. So it's not just full-on segmentation, but it can also be used to assist the clinical workflow. Connection to my research, I go over this a little bit faster. I think I've been talking too much already. One of the problem we were trying to address was that the MetSam was struggling with subtle boundaries. It had poor contrast between lesion and background. For the images who had poor contrast between lesion and background, it was not doing as well. And there was a need for test time dominant adaptation, as we talked about. The approach that we took, we were using SAM at the time when we published. MetSam, I think, was published maybe a couple of weeks before our paper was published. So we combined SAM with level set active control models. What we do through the pipeline is that we first initialize the contour with SAM segmentation, very similar to the clinical workflow that we talked about. So unseen data, we initialize contour with the SAM segmentation. Then we refine the boundaries, but instead of doing it manually, we do it through the energy minimization and through the active contour model that we have. And again, this approach is taste sound adaptation, so it's fine-tunable on small subsets of target data. We evaluated on 4 different data sets. One was ISIC, or the skin lesions that you see the results here, and the other one was the retinal vessel images that I have not included results for this to move faster. What we saw was that we had got IOU, or intersection over union, improvement from 0.64 to 0.75 for the segmentation. Here you see some results, and the precision improved from 0.87 to 0.96. Also on the retinal vessels, which Sam was struggling on, we also got an IU improved from 0.8 to 0.37. And it was the work that we did, it was particularly effective for skin lesions with distinctive boundaries. Very quickly, we do have slides, but I go over this quickly. The recent work that I've started was during the past 5-6 years of my research. There's a lot of foundation models coming out, transformers coming out, image segmentation, but all of this, they're all depending on a large set of training data sets. And when they want to tackle the unseen data, it's as if they have learned nothing, and they don't quite look like human does. And even I'm not an expert, but if you show me some of the segmentation tasks, I might do better because of the stuff that I've seen. So what we're trying to do is trying to learn from human visual patterns, and we're trying to integrate radiologist gaze data with the models that we have. Again, I forgot to include the pipeline. But for the gaze data, we have the gaze data of 3 different radiologists, and through the segmentation task, we are trying to use their gaze information, exactly like we have the prompt encoder in SAM, as a guideline for the model to learn where to look at. And our hope is that this will make the model learn some of the landmarks from the image, and the landmarks would be transferable, unlike the weights in the deep learning models. And the performance assessment that we have had so far was that the model is learning in the gaze patterns would go through the attention of the model would go through the gaze patterns as similar to the clinicians. But again, we're completing the work. If anyone would like to talk further about it, please do reach out to me. The current limitation, I've talked about it already. There's a modality imbalance for MetSam. In general, we do have more images of CT, MRI, and endoscopy. We have difficulty with vessel-like branching structures, like retinal vessels, and there is bounding box ambiguity when multiple structures share the same space. My approach's limitation is, one, the active control performance depends on initial control quality highly. So if yes, I will finish quickly. We're trying to improve the active control models and the hyperparameters tuning. For the gaze model that I mentioned, again, we're trying to see if we can get rid of the variable radiologist gaze patterns. And again, both of the approaches that I'm taking, they have computational overhead for the pipeline. We're trying to integrate the approaches that we have with MetSam to make generalizable model and to come up with test time adaptation. For the clinical translation, we're optimizing for real-time interactive segmentation. We're trying to have a personalized model based on individual radiologist patterns, integration with clinical workflow systems, as was mentioned in MetSam, and having an educational tool for training radiologists. The technical enhancements is extending to true 3D segmentation, not just slices, and having a multi-model fusion. Conclusion, I am out of time, and thank you for your attention. If you have any questions, you can also reach out to me to my email. Thank you.
All right, thank you, Noor. Okay, so we have one more from Stephen. So Stephen, maybe, yep, go ahead. And Stephen will talk about foundation models, particularly around lung cancer screening.
Hello, everyone. My name is Stephen Park. I'm a second-year bioengineering PhD student at UCLA and also working with Dr. Xu. So I'll be presenting a paper that was published this year in February called Medical Multimodal Multitask Foundation Model for Lung Cancer Screening. So as you might know, lung cancer is the leading cause of cancer-related deaths. And lung cancer screening has been shown to reduce the mortality rate of lung cancer. And artificial intelligence methods have demonstrated promise to improve the quality and efficiency of lung cancer screening. However, the problem is that previous lung cancer screening-related AI models have been developed with relatively small single modality datasets for individual tasks, which limits their generalizability to multiple clinical tasks that may be relevant and making them more prone to shortcut learning, which means they learn signals that are not clinically significant. Also, most of these models require bounding box annotations, for example, bounding boxes of the nodules, which makes a large-scale training data sets very infeasible to curate. Hence, the aim of this study was to develop a scalable multimodal multitask foundation model for lung cancer screening, and this was named Medical Multimodal Multitask Foundation Model, so M3FM, by the authors. So this is the overall framework for how M3FM works. So you'll see that the main inputs to the M3FM models is the CT image, the region of interest in the CT image, as well as clinical data, which is converted first into this clinical text, as well as questions that the user wants the model to answer. So, for example, are there lung nodules, or predict the lung cancer risk over 6 years? And you'll see on the top that the model spits out answers to these questions based on the inputs. So, for example, for predicting the lung cancer risk over 6 years, it would produce the risk scores over the 6 years as seen here. In terms of the data set they used to train and evaluate their model, if you see the figure on the right, you'll see that they used a lot of data sets and a lot of different tasks, but ignore that for now. to summarize the data set, there's about 163,000 chest CT series that they used in total, and 49 unique clinical data types, including patient demographics, smoking history, and pathology results. And they also have labels for 17 different lung cancer screening related tasks. And those are in the figure on the rightmost. Examples of these are, again, lung cancer risk prediction, nodule size prediction. And it's not limited to only lung cancer, but also cardiovascular diseases, as well as COVID-19. So those that can be seen on the chest CT. In terms of the M3FM architecture, just to provide a high-level overview, the main components are as seen in the figure on the right, the CT vision transformer, which encodes the imaging data and produces image feature embeddings, and then the text transformer, which converts the clinical text and the questions into, again, text feature embeddings. And those embeddings would move on to the task encoder, which does the multimodal integration and also produces a task-specific feature vector. And then this would be moved on to the task-specific predictor, which you can think of as like the classification head, where it will produce the results, the answers to each of the task-specific questions. So in terms of the training strategy, again, a high-level overview, there are two main steps they took. First is self-supervised pre-training, in which they used the masked autoencoder method for both modalities. So they mask out image regions, and for text, they mask out different text information, and they try to predict the missing information from each of the modalities. And then the second is multitask learning, in which they introduce a method they call a distributed task parallel training strategy, in which a separate device, GPU device, for example, is allotted to each of the tasks, and each device would perform its specific task, and the weights of the model would be shared across the different devices. And hence, the gradient would be accumulated, and then it would do a joint training along these multi-tasks. In terms of the evaluation analysis, they, of course, evaluated the classification performance across their different tasks. And the baselines they compared against were task-specific models that were developed previously. And these were trained based on the task-specific data. And in terms of the metric, they mainly present its AUROC. And the second analysis is ablation studies, where they compare single modality, single task training, as well as multimodality, single task training, and then their own model, which is multimodality, multitask training. And this is to see if each of the components is helpful for their model. So moving on to the results now, first starting with the performance compared to baselines. If you see the figure that's shown, the x-axis is showing the different tasks, and the y-axis is showing the relative improvement in percentage in relation to the orange bar, which is the previous state-of-the-art model based on the task-specific, based on the specific task. And you'll see that across the different tasks, in general, the M3FM improved the prediction performance, as you can see in the blue bar, and they also compared against GPT-4o. And yeah, generally, M3FM improved the prediction performance by up to 20% across the different tasks. And then another aspect of AI modeling that's very important is explainability. And the authors attempt to do this by producing attention maps for both imaging and text that highlight elements that are important for prediction. So if you see the figure on the right, which is for the particular task of cardiovascular disease diagnosis, you'll see that in the image, the regions that were deemed important by the model are highlighted in red. And it is in like the heart region of the chest X-ray, chest CT. And for the text as well, words like diabetes, heart disease, heart attack, and hypertension are highlighted in red since they were considered important by the model for the prediction. In terms of generalizability in an external data set, which is also important in AI modeling, M3FM, their model performed better than the baseline models across multiple tasks in an external data set. And you'll see that they compared against ClipCT, which is another vision language model, as well as Sybil, which is a very popular cancer risk prediction model. And they also did ablation studies where they compared, again, single modality training and single task training with their own model. And you see that M3FM performed better than each of the different baselines that they compared against. Another benefit of foundation models, as mentioned earlier, is that they're able to be used as a baseline for different tasks. So transfer learning to out-of-distribution data is an important aspect of foundation models. So the authors fine-tuned M3FM to predict immunotherapy-induced pneumonitis from CT using a small out-of-distribution data set. So this wasn't included in the training. And you'll see on the figure on the right that M3FM achieved a 4.7% improvement over the competing models that they compared against. And the reference again is the state-of-the-art model that was trained for the specific task. So some discussion on the key results. M3FM offers a framework that handles multimodal data and performs multiple lung cancer screening related tasks. And M3FM outperforms state-of-the-art models across the different tasks. And again, these state-of-the-art models were trained on the specific task. And M3FM was found to be generalizable to external data sets and adaptable to out-of-distribution tasks with limited data for fine-tuning. So some limitations and opportunities. In terms of limitations, I found that the model relies heavily on the creation of large-scale multimodal, multitask data sets that also require labels. And this is, as you might know, very hard to obtain in the medical imaging as well as the whole medical domain. And I found that the question structures are predefined. So one question I had is it flexible to like other like free text type of inputs instead of structuring it in their own predefined way? And some opportunities is expanding to tasks predicting other abnormalities in the chest. So not only in the lung and the heart, but other abnormalities that can be found in the chest, CT, and also fine-tuning for adaptation to out-of-distribution tasks. In terms of some connection to my work, a lot of my work focuses on multimodal fusion of imaging and molecular data, for example, blood tests. And the figure on the right is showing integration of CT and methylation profiles. So one thing that can be benefited from foundation models is that we can address the data size limitations by extracting more predictive and robust features using these pre-trained foundation models for each of the different modalities. And then the second is multimodal representation learning to extract more biologically meaningful imaging features. And the idea is to fine-tune these pre-trained imaging foundation models using multimodal imaging and other data modalities to guide the model to learn more biologically meaningful and more enriched features that can be learned from other modalities. Thank you for listening, and feel free to contact me with any questions. Thank you.
That's
Great.
great. Thank you all, William, Stephen, and Noor, for the great presentations. So I think we can begin the Q&A sessions where I'm going to read questions either from myself or from the audience. So I'm going to begin with the question with the paper that William presented. So what ethical considerations arise when using AI to estimate biological age, and particularly in scenarios like insurance companies and employment screening?
Yeah, absolutely. I think definitely this is a concern, right? Because I think there's several layers to this question. One is, whenever we make these type of models, because certainly just because a machine learning algorithm can do it doesn't mean you can, you want to just build the model and apply it without really understanding, number one, is it making the right prediction? I think there's a lot of benefit in potentially being able to use things like imaging as the basis for risk prediction, partly because right now a lot of the risk prediction is done using clinical variables, right? Things that, for example, for women going through breast cancer screening, there's Tyrer-Cuzick and these kind of clinical models that, use very coarse kind of variables that an individual may or may not even remember correctly to make a risk prediction. And imaging could be a more objective basis for doing that. But that said, I think when what you just mentioned about, the ethical considerations about, how this is being used, also, the transparency by which we are building these type of models, right? I think certainly the UK Biobank was a large, informed consent type of study where, clearly the participants knew going in that their data would be used for this type of research. But ultimately, because we in our health systems, a lot of patients, we get data, we do need to make sure that there is clear transparency about how the patient's data is being used. So I think maybe, it's a very important question, and I'm probably just touching a little bit on it, but I do think that there are definitely concerns of prematurely using these type of models without really the safeguards of, number one, fairness and transparency, number two, really just being able to apply it in the appropriate manner, right? Because models will give you a prediction regardless of whether it's correct or not, and that can really have strong downstream implications.
Yeah, thank you so much, William. This is great insights. And I think it would be very unfortunate if things like some insurance companies that use LLMs to deny patient claims, that kind of things happening again with this kind of model. So the second question, I think, goes to the paper that Stephen presented. And this one more technical question. So how did the authors address alignment between different data types? Because there have been different asynchronicities of the data types and frequencies, like EHRs might be more frequent, and then the images might be less frequent. So how does the alignment, if any, did the author perform?
Yeah, so the alignment is basically performed by their, as I mentioned, their distributed task parallel training strategy, in which they train these different task-specific devices. So they allot a model to a certain GPU device. And this particular device can take in any types of clinical data. So like imaging only versus clinical data only, or imaging plus clinical data 1 plus clinical data 2. But this structure is consistent in this particular device, and they address a specific task. And then these different devices with different data inputs and different combinations of data inputs and different tasks are accumulated, and they accumulate the gradient and do the back propagation and train the model that way. Yeah.
So maybe I'll just
Thank
add
you.
one more thing about Stephen's answer. I think he answered from a very technical perspective, but I think, Jan, maybe also what you're getting at is, how do you correlate like the imaging, the text, right? If you have a lot of this data, right now, for those of you guys who are familiar with like clip models, right, they're basically looking at similarity between the mentions of the text and parts of the image. But I think to your point, I mean, this is a big needle in the haystack kind of problem, right? You have very small nodules, but then you have all this text and the large high dimensional images. And so I think one of the opportunities I see about trying to train better foundation models is really better ways at getting at the, helping the model at least learn the relationship between these two. And I think that's still a very much open area, and happy to chat with people about collaborating in that space.
Yeah, that's great. And thank you so much for the well-rounded insights. And I see that we are at the top of the hour, and due to the limit of the time, we're going to have a very short Q&A sessions, but please feel free with our to follow up with our presenters if you have any follow-up questions. And I want to thank our presenters again for their wonderful presentations, and I especially like that they draw the relation to their own work. This is kind of example takeaway message that we want the audience to also get. And so I want to thank all the presenters again, and then remind everyone that we're going to gather again in 1 month. And in our next session, we're going to be featuring our friends from Emory. But today, we're going to thank our UCLA friends again. Thank you so much.